Welcome to Sustainability Soundbites, presented by E&E Leader. UT/MD Anderson trace microplastics from food to cells. 1 year into a joint research project, scientists at UT Austin and MD Anderson Cancer Center say they are seeing measurable biological changes in animals exposed to food-grade microplastics well before any tissue damage becomes visible, adding a new dimension to open questions about plastics and human health. Researchers at the University of Texas at Austin and the University of Texas MD Anderson Cancer Center reported preliminary evidence this week that microplastic exposure can produce measurable biological changes before tissue damage becomes visible under a microscope. 1 year into the Empathic Project, short for Environmental, Microplastics, and Systemic Pathology, Inflammation, and Carcinogenesis, lead researchers Andrea Viale of MD Anderson and Jianfei Liu of UT Austin say animals exposed to food-grade microplastics show changes in metabolism and other biochemical pathways, even though their organs still appear normal under the microscope. The findings do not establish that microplastics cause cancer. Instead, they indicate exposure may trigger biological responses before more obvious physical changes occur. And the project is now examining whether those responses persist under longer exposure. Following microplastics from environment to tissue, Empathic's distinguishing feature is its attempt to connect environmental exposure with biological outcomes rather than studying either side in isolation. Liu's team at UT Austin identifies and characterizes microplastics found in food, drinking water, and waterways, using pyrolysis gas chromatography mass spectrometry to quantify particles and characterize the additives they carry. Viale's team at MD Anderson then investigates where those particles accumulate in tissue and whether exposure produces molecular or cellular changes tied to inflammation or cancer. Complicated by the fact that microplastics are not a single contaminant, but a broad category varying in polymer composition, size, shape, and surface traits, variability that has similarly stalled Rhode Island's proposed statewide microplastics ban, which needed its own testing framework first. Measurement remains the central obstacle. Before determining what microplastics do inside the body, researchers first have to reliably determine whether they are there, and standardized detection methods for biological tissue remain limited. During its first year, the team developed new detection protocols, built shared analytical workflows between the two institutions, and created animal models approximating human exposure through food and drinking water, along with platforms to study potential effects on pancreatic, liver, colon, and lung cancers. That groundwork could matter beyond cancer research, since reliable measurement is what separates a genuine biological signal from analytical noise, the same detection gap that has complicated earlier findings linking microplastics in human blood to cardiovascular risk. Earlier cancer research set up the next question. The project's cancer hypothesis builds partly on Viale's own earlier work. In a 2021 study published in Science, Viale and colleagues found that pancreatic tissue can retain molecular changes after inflammation appears to have resolved, a persistent reprogramming the researchers called epithelial memory that can influence later tumor development when combined with oncogenic KRAS mutations. Empathic researchers are now asking whether repeated microplastic exposure could leave a similar imprint. They have not established that it does, but the observation that biochemical changes occur while tissue still looks normal gives the team reason to keep investigating longer-term exposure. For companies in plastics, packaging, food production, and water management, how much this research matters depends on whether scientists can connect specific types or levels of exposure with specific biological outcomes, a link the current findings do not yet provide. If future research does make that connection, distinguishing among polymer types and exposure pathways could become relevant to environmental monitoring, product design, and eventually regulatory risk assessment, a bar similar to the one facing chemical classes like PFAS, where measurement and liability frameworks are still being built compound by compound. For now, Empathic is working through a more basic problem, tracing particles from environmental sources into biological systems, since the early findings suggest waiting for visible damage may not capture the full response. Interested in a soundbite sponsorship? Check out the energy category, currently sponsored by Amazon. Reach out to our team for more information. Thank you for listening.